called me Edward Slide Hands. Hi guys, and welcome to another virtual lab. Today we are actually going to be making slides with garlic cells and cheek cells. So I'm going to kind of take us through everything. I'm going to show us how we isolate the cells from the garlic as well as from our cheeks. We're going to stain them and then we're going to look at them underneath the microscope. So without further ado, let's go ahead and get started. So the first thing I need to do is I need to put on some gloves because we're going to be working with hydrogen chloride, which is an acid that can hurt your hands or your skin if it gets on your skin. So we got to make sure that we are being safe. Now, I'm only going to use a few drops of it, but even still, I got to make sure that I'm following safety protocols. So right now, what I'm going to do first is I have to dilute my hydrogen chloride because I only need it to be at a molarity of 1, but the hydrogen chloride that I have in my classroom is at 3. So that just means that I'm going to dilute it 2 times. So what I'm going to do is pour it out to the 10 milliliter mark. Okay, so I've got it at the 10 milliliter mark. And then what I'm going to do is I'm going to use some distilled water and then pour it to the 30 milliliter mark. So it's 1 part of the hydrogen chloride and then 2 parts water, which means it'll be diluted from a 3 to a 1. Oh, that is right there. And this is more than I need, but that's okay. I can save some for the next lab that we're going to do. So that's there. Now what I'm going to do is I have my little bundle of garlic. Let me go ahead and get it separated here. Forgot I have a trash can next to my table. I moved things around in my classroom a little bit since we'll be recording more labs. So I'm gonna switch things up a little bit. The procedure that I found online said to use the little roots that would form at the bottom of the garlic or the onion sprouts that you might have, but I wanted to see what it would look like if I used the garlic flesh itself. So we're going to make a few different slides. So speaking of which, I've got my slides here. I'm going to separate them for the... I'm stronger than I look. Okay, I think we are good with the number of slides I have laid out. Just gonna open the box for the slide covers to get that ready. So, first thing we're gonna do is I'm going to take my little roots here. Let me show you nice and close up. So these are the roots that you would find at the base of like a thing of garlic, or if you have onion roots, the green onion, all of them will form these little roots. And as we now know, mitosis is the way that our cells grow and replicate, which means that there's a very high chance that we can catch some cells doing mitosis actually in these roots themselves. Just because this is where a lot of growing and replicating is going on. If you think about it, cells need to grow so that the roots can get nice and long in order to access nutrients and water in the soil. So if you have garlic or onion at home and it doesn't have little roots growing off of it, then what you can do is put a few toothpicks in the garlic or the onion and then have just the base submerged in the water, and then very quickly some roots will start to emerge to take advantage of that water. So I have my How on earth did that happen? I wonder if my roots just flew off of my sled as I was going to carry it over to you guys. And it fell right into the trash can. Here it is. All right, so I'm gonna take a picture of all of these because obviously I can't move it to show you because I just tried and then I'll flood in the trash can. So I'll insert pictures of what all of these look like as I prepare them. This is why I need a second camera that shows exactly what my hands are doing. I'll get that set up soon if I have to keep making more of these videos, that's for sure. Okay, so I've got my roots out. What I'm gonna do now is I'm going to do 2 or 3 drops of hydrogen chloride, which remember I had mixed up here. So I want to make sure that all of this hydrogen chloride is mixed with those roots, and they're just going to sit there and do their thing. And that's just going to wait for 5 minutes. So let me set a timer here for 5 minutes. And what this hydrogen chloride is doing is it's essentially just weakening the cell walls, weakening the cell itself, so we can get access to the cells enough to get some pictures of what's going on in there, because once again we want to catch it doing mitosis. So I have my clove of garlic here, and it actually looks like there's some cell replication going on at the base. I can see some new sprouts emerging. So I'm going to use my tweezers here, my forceps if you want to be more scientific, and try and just cut off a small amount. And I'm going to put it on another slide and do the same thing. I'll do a few drops of hydrogen chloride and we'll wait for 5 minutes. Let that do its thing too. So now what I'm going to do while we're waiting for those to soak, I can take off my gloves because I'm done using the hydrogen chloride. For the moment, I'm going to get a new pair of gloves when it's time to blot it. Probably should have kept my gloves on while I moved that. So now what I'm going to do is I'm going to use a toothpick in order to get some cheek cells for the cheek slide. So got my toothpick. These ones are nice, but one end is pointy and one end is blunt. That way I'm not going to stab myself in the cheek. And so what you do is you just gently scrape it. You don't want to like try and gouge a hole. You just want to gently scrape. So that's what I'm going to do. As I was scraping, I was kind of rotating around just to increase the chance that I got some cells on all parts of the stick. You can't really tell much on the cell that I used my cheek cells for. That's because it just looks like spit. But there might be some cheek cells in there. So we're just gonna let the hydrogen chloride do its thing. I'm gonna go get some more gloves and get some paper towels so that we can blot off the extra. And then we can jump right into the next few steps. I have my paper towels and I have my gloves. So now what I'm gonna do is I'm actually gonna start staining my cheek cells because that's gonna take about 3 minutes for them to fully absorb the stain. And what I'm using to stain my cheek cells is a substance that's known as methylene blue. And what methylene blue does is it actually binds to our DNA and RNA, and because we're trying to see if we can find cells undergoing mitosis, remember DNA, when it forms and condenses into the chromosomes, that is one of the main things that we're going to be able to see, hopefully, because it's just a bunch of DNA, which means that the methylene blue should stain it, and we should be able to see it nice and clear. So what I'm going to do is pour a little bit of methylene blue out here. Another reason I'm wearing gloves is because methylene blue does stain quite a bit, so I want to make sure I'm not getting it all over my hands. Okay, so I have just a little bit here because we don't need that much. What I'm going to do is just put a few drops over the slide with what I hope are some cheek cells. There goes my timer. Perfect timing for us to soak up this hydrogen chloride. Let me set the timer for 1 minute because I remember I need to do an extra minute for the actual part of the garlic. So what I'm going to do is just blot up the hydrogen chloride. so that we can stain it with the methylene blue. So I'm not even going to put the paper towel directly on top of the parts of the roots, just because I don't want them to stick to the paper towel. If we remember properties of water, cohesion and adhesion, if you just put the paper towel close enough, it'll actually soak up all of that water without you having to like move it around. It just pulls it all up into the paper towel. That's how we were able to do the chromatography lab. Now that I have all of the hydrogen chloride soaked up, what I'm going to do is take another pipette. I know lots of pipettes, and we've got to make sure that they're all clean. I'm going to get some deionized water. And essentially what this means is that it doesn't have any ions in it that might otherwise interact with the other substances that we have at work here. So before I was able to do that, my timer went off. So let me go ahead and soak up the extra hydrogen chloride from my other slide here. Perfect. So now I can just do them both at the same time. So what I'm going to do is take my deionized water and I'm just going to rinse off any excess hydrogen chloride. Okay, so we're going to do a few washes of this. So that was my first wash. And then let me do my second one. Now that I've got those nice and rinsed off, we're going to administer some drops of the methylene blue. So I've got that right here once again, and I'm just going to do a few drops so that we can get them stained and then hopefully be able to see some cells undergoing mitosis. Now the chance that we'll see cells undergoing mitosis is higher in the garlic root cells than in my cheeks, just because of the fact that they're on the tips of those roots there, which means that they're probably undergoing much more active mitosis cell replication than the cells in my cheeks, but thought it would be worth a try since I've got all of my equipment out anyways. So what I'm going to do now is I'm going to soak up the methylene blue from my cheek cells, soak up any of the excess, and then I'm going to rinse off my cheek cells with some deionized water so that all we see that stained blue is just going to be cells and the parts of the cell like DNA that we are trying to see. I'm hoping I don't absorb up all of my cheek cells in the process, but we will know when we try and look at it under a microscope. So I put some deionized water down, and then what I'm going to do is put a slide on top. It's a slide cover here. I've got this slide prepared while the methylene blue soaks the cells. Let's go see if we can find some cheek cells under a microscope. Alright, so I was able to see my cheek cells. I'm going to make another slide because I didn't see any cheek cells undergoing mitosis, and like I said, I've got all of my supplies out here. It's not very difficult. Apply it to the slide here. Same exact process. I'm just kind of putting the toothpick down and putting some methylene blue over it just to stain what I'm hoping are some cheek cells that are undergoing mitosis. That is the goal. That is what we're looking for here. I think we are good to soak up all of the excess methylene blue from our garlic cuttings here, little samples. So we're going to kind of do the same exact thing that we did with the hydrogen chloride in terms of we're going to rinse it off, some deionized water, and then see what we can find under the microscope. Now what I'm going to do is I'm actually going to separate each of the roots to a different slide so I can look at them one at a time. And then I'm going to also break up the little cutting that I had made of the garlic itself to see if I like those a little bit better.
I just wanna be.
The last thing that I'm going to do here is I'm going to take my slide cover. I need a few of them since we've got a few slides here. And then the last part of this is to actually crush the roots enough to manually break apart those cell walls so that we can really get a glimpse at what's going on inside those cells.
I just wanna be.
Good cheek cells on this one, I can feel it. All right, let's go ahead and look at all of our different slides under a microscope. They called me Edward Slide Hands. So that is the lab. I hope you enjoyed watching me put together those steps so that we could see some cells undergoing mitosis. If you guys have any questions at all while you're completing the worksheet, please let me know. Otherwise, I will see you guys soon. Thank you. Edward Slide Hands.